Good day to everyone joining us, and welcome to today's webinar featuring Bend Research, the role of amorphous dispersions in progressing poorly bioavailable compounds from discovery to commercial product. This presentation will run for about 20 minutes, including a Q&A session towards the end. I would like to now welcome our speaker for today's presentation, Dr. David Lyon. David Lyon is a Senior Vice President at Bend Research, Inc., where he leads efforts in scientific excellence and global business development. His expertise includes solubilization of pharmaceuticals, nanotechnology-based drug delivery, and controlled release formulations. Dr. Lyon joined Bend Research in 1991. He holds a BS in chemistry from Western Washington University and a Ph.D. in inorganic organometallic chemistry from the University of Oregon. And now, without further ado, here is David Lyon.
So a challenge facing the pharmaceutical industry currently is really around the increasing number of compounds that are classified in the Biopharmaceutics Classification System, or the BCS system, as BCS Class 2, where solubility is really the driving issue. And you can look at a number of solutions that are provided from the literature and even commercial products where pharmaceutical compounds are formulated as salts. They can be made as solutions, micronized, or formulated in lipids. But really, driving forward, there's been a need over the last decade for technologies that can increase solubility and drive bioavailability. And today's talk is really going to focus around the use of amorphous dispersions to get these BCS Class 2 compounds bioavailable. When you're talking about developing an amorphous dispersion for the pharmaceutical industry, really you have to consider all the way from discovery through commercial. There's a lot of ways you can get highly performing, that is, highly bioavailable amorphous dispersions to PK studies, pharmacokinetic studies in animals, or even to the clinic. But really, a critical issue that's not often appreciated is being able to not only drive that performance, but also get that performance to where it's manufacturable at a commercial scale, it is a stable formulation to commercial scale, and also has been integrated into a dosage form in commercial. And so this slide 3 really shows, ideally, that you'll consider all these things early on. But reality often is that rather than an integrated approach where your IND-enabling formulation needs to be commercial and it's fast to market, often because it's a first-in-class compound, often the early-stage investment in a compound is minimized, and fit-for-purpose formulations are used preclinically or in early clinical, so-called fast demand. But then when you get past the proof of concept study, you often end up in a rescue mission, and so reformulation is necessary to account for often increased dose, decreased stability due to dose and process, compromises in a dosage form, something that was fit for purpose, or even the need for a new technology. Amorphous dispersions can be manufactured by a number of different processes. What's really out on the market right now and what's available from early clinical through commercial is a spray drying process, which is shown on the left hand of the slide, where a solution of the active pharmaceutical ingredient is dissolved with a polymer and a common organic solvent. It's then spray dried, removing the solvent, and resulting in a homogeneous, stable amorphous dispersion when appropriately formulated. Alternatively, a non-solvent process is the hot melt extrusion process, where a drug and polymer is blended and then fed into a hot melt extruder. The combination of the temperature and the mixing of the extruder is then used to form the amorphous dispersion. This is less applicable due to the need for a thermodynamically homogeneous dispersion as opposed to a kinetically stable dispersion that you can access with a spray drying process. Bend Research has been involved in formulating amorphous dispersions for well over a decade now and have seen significant portions of the pharma pipelines. More than 700 compounds have now been progressed into through feasibility, in vitro feasibility assessments, with more than 450 advancing through preclinical animal in vivo studies. And then more than 65 have now been advanced through clinical trials as well. And this has resulted in a few commercial products that are formulated as amorphous dispersions. Representative clinical data is shown in the lower left-hand corner of the slide. And generally what you see is that an amorphous dispersion, whether it's spray-dried or manufactured via hot melt extrusion, provides a significant increase in the AUC in the clinic, and there are 3 examples from phase 1 through phase 3 representative data, or in the preclinical arena as shown in the lower left. Just to make the point that this is not limited simply to specific therapeutic areas or even specific types of compounds, in the lower right-hand corner of the slide, you can see that there's an example chemical property space in which we've plotted a melting point over a glass transition temperature of a compound, which is simply the driving force for crystallization, against the log P, which is really about the lipophilicity or the dissolution rate of a compound. And you can see that we've seen and formulated a wide variety of compounds. In fact, it's very much a shotgun spray across the chemical property space, showing that amorphous dispersion technology is quite applicable to a wide variety of physical-chemical spaces. When formulating as an amorphous dispersion, it's important to be able to be predictive of the performance, the stability, and the manufacturability of a compound. And page 7 shows a variety of different spaces, and again, in which we're showing the melting point divided by the glass transition temperature on the y-axis versus the lipophilicity of the compound, or the log P of the compound, on the x-axis. And you can see some challenges arise when you hit the extremes of this type of plot. If you have a low melting, high glass transition temperature compound, so a low propensity to crystallize, you find that the compound is quite stable, and high loadings, that is high drug content in the polymer, are quite possible. As you increase the melting point or decrease the glass transition temperature, there's a tendency for crystallization, and that leads to physical stability challenges. And so those can be formulated around, but it's important in the spirit of being predictive of what your formulation will do to identify that early and not just have a empirical approach to the formulation, but in fact have a predictive way to determine the formulation. Similarly, if you have a high log P compound, it will tend to have poor wetting and a poor dissolution rate, and the loading, again, can be limited, but also can be predicted based on the compound's properties. So slide 8 then shows both the spray-dry dispersion SDD performance and its predictability. In the upper left-hand corner of the slide, there's a graph of the plasma concentration versus time, so the PK curve, for compound A, which was dosed at 200 milligrams active in the fasted state. And for a 25% dispersion, it's clear that for this compound that has a low solubility, a 1.3 microgram per mil solubility, non-ionizable, and a modest log P, that the SDD, or the amorphous dispersion, gives a much higher area under the curve than the crystalline drug. In the upper right-hand corner, then, is the SDD, which was dosed as a powder in a bottle, compared to a tablet. And really the key message here is that it's possible, and in fact quite easily done, that a tablet can provide the same exposure as a powder in a bottle. And so for early clinical trials, it's often not necessary to formulate a solid dosage form, but it can be, the SDD can be formulated as a powder in a bottle, which decreases costs and decreases the timeline necessary to get to the clinic. And then finally, in the lower quadrant of the slide, it's not only important to recognize that the spray-dry dispersion can perform well both as a powder in a bottle and a tablet, but it's predictable. And so in the lower left, you can see that there's a guidance surface of what the dose absorbed for compound A should be for a crystalline compound. And that's plotted as a function of the log P, again, the lipophilicity of the compound, and then the solubility or the free drug concentration of that compound. And not surprisingly, the prediction from this biomodel suggests that this compound A would be poorly absorbed as the crystalline compound. And in fact, as we've already discussed, the crystalline drug is poorly absorbed. However, if the prediction is then made, not changing the log P, of course, because that's a physicochemical property of the compound, but the aqueous solubility, which can be increased due to the amorphous form of the compound in a dispersion, but also the dissolution rate, which is also increased in the amorphous form, one predicts that the compound will be well absorbed, 70 or 80% absorbed, as shown in the lower right-hand quadrant of the slide. In fact, that's already a result we've discussed, and the compound is well absorbed as an amorphous dispersion. So again, the summary of this slide is that the SDD does perform well, and the performance is readily predictable. So slide 9 now talks about the mechanistic models for dissolution of SDDs. And really there's two fundamental mechanisms that need to be considered when formulating a compound. One is one where the drug has low solubility in the hydrated polymer, and then upon hydration, so as it goes into the gastric media, it nanophase separates into nanostructures or nanoparticles that then come off of the parent amorphous dispersion particle, rendering a very rapidly dissolving and high energy form. Alternatively, when a drug has high solubility in the hydrated polymer, it tends not to nanophase separate, and in fact an erosion mechanism occurs. And really the key point to make here is that depending on which mechanism is fundamentally operating, the formulation may change to account for the different dissolution rates and the different dissolution mechanisms. So again, back to predictability, to aid formulation and to aid the speed at which a clinical trial can be conducted and in which a program can be accelerated, we've developed a oral delivery biomodel. And really the key thing to note here is that with a few physicochemical properties, that is what is the true solubility of the compound and what's the enhancement of that solubility in the amorphous form. If you know the micelle partition coefficient, the dissolution rate, and then how much drug stays in the nanoparticle or in colloids, by taking a pretty simple continuously stirred tank reactor model where we model the duodenum, a couple of jejunum compartments, and an ileum compartment, you can take a flow rate into a compartment and flow rate out of the compartment, the dissolution of the drug, whether it's as the crystalline drug or as an amorphous dispersion, and then finally an effective permeation rate, which can be calculated based on the molecular weight and the log P of the compound. Then those predictions can be very accurate for the amount of drug that would be absorbed as the formulation passes through the GI tract. And then slide 11 simply shows that for a variety of compounds, we've plotted the observed fraction of the dose absorbed as a function of the predicted fraction of dose absorbed. And you can see that from this graph that there's a fairly good correlation, well, not perfect, because we're not taking into account all of the things like metabolism or gut metabolism that might occur due to other things going on. We're simply looking at fraction of dose absorbed, and in this case, it's a very good prediction. So early on in the development program, some very simple physicochemical properties can be taken into account, and predictions can be made on what kind of improvements we will see in absorption. So another key piece of the formulation is not just the compound, but the excipient that you might use in formulating that compound. And in some work we've done with Dow, and there's a companion presentation during this virtual bioavailability webinar, looking at optimizing the excipient for performance. And you can see in the case of hydroxypropylmethylcellulose acetate succinate, or HPMCAS, that there's some specified grades in the USP that aren't accessible using kind of a commercial grade. And so with Dow, now there's the availability of a variety of grades of polymers outside of the commercially available grades, and those are now available to do quality by design, or QBD sampling. And so some of these grades are shown on slide 12, and just very simply on slide 13, the performance for 3 modeled compounds as a function of these different substitution patterns has been evaluated. And so in the upper left-hand corner, we've done a microcentrifuge test looking at the amount of drug dissolved in solution plus the amount of drug in colloids, or in nanoparticles, as a function of the substitution pattern. And the other 3 graphs on the slide show that for some compounds like itraconazole, it's fairly insensitive to the substitution pattern, but for a compound like phenytoin, which is a very greasy compound, or torcetrapib, which is shown in the lower right-hand corner, the performance is quite sensitive to substitution. And this is another handle, other than just simply changing grossly the polymer or excipient composition itself. Just some subtle changes in the excipient can be used to improve performance or optimize performance, or even if the commercially available grade is desirable, look to the edge of failure for performance in the commercial filing. So once performance has been defined, it's important to also know that your compound or your formulation is going to be physically stable. And it's important, I guess, to use a phase-appropriate physical stability testing. That is you don't want to make too big of an investment too early on a compound that won't progress, but also having the robust and scientifically rigorous data that's going to be convincing the filing that the amorphous dispersion will be stable long-term. And so what we've developed over time is in early phases, so as you go from screening even to the early clinic, is a rule of thumb based on the glass transition temperature versus the storage temperature. This, as you get into the clinic, is then overlapped with some physical stability mapping, where you start looking at some purposefully crystallized material. And then finally, in the real clinical phases, of course, there's ICH stability data that's taken into account. And so the phase-appropriate physical stability screening that we do in early phase, as I said, is based on the glass transition temperature. And so as you look to physical changes as the SDD is stored at a glass transition temperature similar to the storage temperature, but then as you get farther and farther away, it allows for a longer-term stability. And so if you look in the graph on page 15 then, for a sample stored of this spray dried dispersion stored at 25 degrees and 50% relative humidity, where the delta is 40 degrees different than the glass transition temperature, it's stable for at least 18 months. In fact, in this case, I believe now we have multi-year stability data. But for the situation in which the glass transition temperature at 4075 is only a modest 6 degrees above the storage temperature, well, we see crystals quite readily. In fact, at 3 months, there's a substantial crystallinity as shown in the SEM in the upper right-hand corner. But this rule of thumb analysis does a really nice job, as you can see on slide 16, in that if we have a glass transition temperature that's greater than 30 degrees above the storage temperature, 100% of the time, the samples are stable. And so just this simple physical measurement in early phase gives us quite a bit of confidence that the sample will be stable. Even when there's 20 to 30 degrees or 10 to 20 degrees of the glass transition temperature is above the storage temperature, then you get still quite frequently stability for all of the samples, well in excess of 80% of the samples. And even when the glass transition temperature is comparable to the storage temperature, at least half the time, for a smaller number of samples, we do have a stable formulation. And so this really gives us the opportunity very early on with very simple measurements to be able to formulate in a way that will be predictive and will lead to a stable commercial formulation. And then finally, of course, on slide 17, it's important to note that physical stability of SDDs is very well established. And here's some data for both spray-dried dispersions in as powders, and again, you can see there's multi-year stability, but also in the dosage form. And of course, with compounds like Excipient now on the market, there's obviously real-time data for commercial products that corroborate the fact that spray-dried dispersions can be stable for multi-years. And just anecdotally, we've cleaned out our lab a number of times in which we've found some samples that have been stored under uncontrolled conditions, and samples that have been over 10 years now have been demonstrated to remain amorphous and have unchanged in vitro performance when they are properly formulated. Then slide 18 really goes to show that 2 points. One is the science of scale. That is that in the early formulation screening or discovery arena, we can formulate spray-dried dispersions and operate really in the discovery constraint where we have tens of milligrams of the active pharmaceutical ingredient available, but that is scalable through kind of grams at our lab spray dryer scale to grams to kilograms at the PSD1 scale, and then finally at Bend Research we have a PSD2 which allows really multi-kilos, hundreds of kilos to be spray dried for late-stage clinical trials. And then, of course, in our announced collaboration with Hobiome, the transfer of these types of formulations to a PSD4 or a PSD5 for larger scale formulations is opportunity and is available. But really, as you scale up, it's important, again, being predictive, to know the physical situation that you're dealing with. And in the upper left-hand corner of slide 19, there's a spray plume for a PSD1, which shows that it's a fairly transparent plume going on to a PSD2 and finally onto the PSD5, where it's a fairly opaque plume. And really what's important to note is that as you think about spray drying and scaling the spray drying process, that as you go from the PSD1 where drying is often not an issue, by the time you get to the PSD2 and absolutely by the time you get to the PSD5, really the isotherm for the spray dryer needs to be taken into account. And so in the bottom of the slide, you can see that the specific drying ratio, so that the liquid flow to the gas flow, or the drying gas flow, versus the inlet temperature of the drying gas is taken into account. And as you go into the upper left-hand corner of that graph, the drying kinetics are slower. You tend to get denser, shriveled raisins. On the other side, if the drying ratio is low, that is that the gas flow is very high relative to the liquid flow, and that gas inlet temperature is high, you get fast drying, and therefore you get balloons, if you will, so hollow spheres is your product. And both of those situations have attributes that can be both positive and negative. If it dries too fast, it tends to have a very low density and needs to be formulated in a way and processed in a way that deals with that density. On the other hand, if it dries too slowly, the particles can be too dense and not really compress well, when they're put into a solid dosage form. So both situations can be dealt with. They just need to be recognized as the process is being designed, and more importantly, as that process is being scaled. Then finally, in slide 20, this is now taking the spray dry dispersion and putting it into immediate release tablet. And the learnings here really focus on providing a maximum surface area of the native SDD particle as rapidly as possible in the upper intestine. And what this really means is going from the macro tablet and dissolving as rapidly as possible in the gastric into the granules, but then fundamentally going from the granule into the primary SDD particle as rapidly as possible. And what that allows is the nanoparticle to form as quickly as possible in the upper intestine and leading to, as rapid as possible, absorption of that drug. If the primary tablet going to the granule, going to the particle, ultimately going to the nanoparticle, if that's slow, what ends up happening is it's more of a matrix tablet effect, that is, it's a controlled release as opposed to an immediate release. And so, again, this is dominated by the drug physicochemical properties, and the earlier you can take this into account in the process and be predictive of the performance, the better off the project will go. And then slide 21 simply shows what the theory said on 20, that is that rapid dissolution is important, and going from the tablet in gastric as a function of time into the intestinal buffer is shown, and this simply says granule equals SDD particle, and finally, when it's put in a tablet, the dissolution rate for all three are identical. So the summary thoughts for this piece of the virtual bioavailability webinar are that amorphous dispersions are an important tool to improve absorption of poorly soluble compounds. The amorphous dispersion formulation can be rationally chosen based on performance stability, process and manufacturability, and dosage form considerations. And the earlier we take this into account in the formulation process, the better off the formulation will perform and the better off the project will go. And then finally, I guess in conclusion, that predictive models and biorelevant in vitro tests can shorten development timelines and decrease API usage. So thank you for joining us today on the bioavailability challenge.
Thank you for listening for today's presentation. Now we'll move on to the Q&A session.
Good afternoon, and we'll now be starting the question and answer session associated with this webinar. We've got a couple of questions coming across the wire here, and I'll go ahead and read the questions to the audience and then have to answer them in the few minutes we have remaining. One of the questions that came across is asking, what is the purpose of your relationship with Dow? As you saw from the previous webinar that was presented by the Dow folks, our relationship is really about getting to new excipients with novel compositions that allow for improved performance in the solubilization arena. And so really it's the combination of the Dow chemical material science expertise and the Ben Research formulation expertise. And that interface allows for both improvement of the excipient as well as the formulation. The second question that's come in, it asks about how we use amorphous dispersions in a discovery setting. And I think this is really important to think back to small scale, scientific scale, and really the ability to use small spray dryers in small in vitro tests that allow you to test at really milligram quantities that are convenient to the discovery chemist. And so the approach we've taken in going all the way back into high throughput screening or high content screening is to be able to manufacture dispersions at the single digit to tens of milligram quantities and be able to test them at milligram scales. And what this feedback allows then is a choice between multiple formulations, choosing the best one to take into a PK study. And so by having that iterative loop where the formulator and the discovery chemist are really talking at that interface, it allows kind of an optimized formulation to be taken into the early PK, the toxicology, and then ultimately into the clinic. We have a subsequent question coming in asking, can we formulate an encapsulated drug product using SDD technology, or is it always a homogeneous matrix where drug and polymer are dispersed together? I'll take the second part of that question, I guess, first. In its typical deployment, both the polymer and the drug are dissolved in the spray solvent for the spray drying process. And so in that sense, those are definitely amorphous dispersions where it is a homogeneous drug polymer mixture. That said, there are a number of technologies, or I would say adjuncts to the technology, where we have looked at suspending either crystalline drug in a solvent, so basically a non-solvent that the polymer is soluble in. And what that allows us is to spray dry effectively encapsulated drug crystals in a polymer matrix. And while they have potential to have high energy characteristics like the SDDs, they also just in some cases are used to prevent crystals from touching other excipients, so to improve chemical stability, or also to allow taste masking to occur, so as the drug and polymer go through the mouth, that it does not touch the taste buds. So I think our time's up for this morning, and I thank you for everybody's attention and questions. And if there are any additional questions, please post those to the Ben Research page for the webinar, and we'll happily send along a written answer. So thank you and good morning.